acidophile. They love acidic environments. So what area in your body is acidic? Well, the stomach. Very few organisms can actually survive in the stomach. Most of them would actually die if they're exposed to the acid for a long time, all right? Anaerobic, anaerobe. So let's go to the one that we can relate first, aerobic. An aerobic organism loves oxygen. In fact, it can't survive without oxygen, right? An anaerobic organism can survive without oxygen. I mentioned Clostridium tetani that causes tetanus. That's an anaerobic organism. In fact, oxygen can kill that organism, all right? A strict aerobe absolutely requires oxygen. Here's another word I want you to remember. This is an important one, facultative. A facultative organism can grow both in aerobic environments and anaerobic environments. Doesn't matter if you have oxygen, if you do, it doesn't matter if these organisms will grow. So you can already tell the most prolific pathogens in humans, they would be facultative. Staph aureus is a facultative organism. It can grow with or without oxygen. E. coli, it's a facultative organism. It can go with or without E. coli, all right? So anaerobes, no oxygen. Aerobe, oxygen, facultative, both. Let's go with free-living organisms. A free-living organism, they're out there in the environment. Sometimes you come in contact with them. Most of the time, they're not causing disease. But if you're in a certain immune state, or you have a propensity to get this disease, or you have some underlying conditions, those organisms can cause disease. Now, the last term I want you to get is this one here, obligate intracellular parasite. To be obligated to something means you have to do it, right? So obligate intracellular, this is a bacteria that lives inside the cell. In fact, obligate intracellular parasite simply means this. These organisms can't grow by themselves. They require a living cell. In other words, they infect the cell, get into the cell, and that's the only way they can survive. All right? So key terms, psychrophilic, anaerobic, aerobic, facultative, and obligate intracellular parasites. The cyanobacteria, we don't have to worry about them. They don't cause disease in humans. They love to be in the environment. They are photosynthetic. Again, if you go to some pond, you see a color on the pond. Maybe it's green or yellow or red. Generally, it's these organisms, cyanobacteria, that are photosynthetic. They have chlorophyll. The pigments have different colors, but generally, these don't cause disease in humans. All right. Okay, so we just talked about bacteria. Next group is Protista. And again, I don't want to memorize too much here because we will talk about this in chapter 19 when we look at parasites. But remember, the only prokaryotes are bacteria and archaea. Archaea don't cause disease in humans. Bacteria do. Bacteria don't have nuclei. All right, so protozoans, these are single-cell organisms. Most of them lack a cell wall. I just want you to know a couple examples. Again, no scientific names or anything yet. Malaria, sleeping sickness. There's one STD that's transmitted by an organism called Trichomonas vaginalis. But nonetheless, these are examples of protozoal diseases. All right? Fungi. What do I want you to know about fungi? Well, later on when we study them, you're going to see there are two groups. One's filamentous, and the other is single cellular. So again, we probably all see this on rotting fruit or maybe some red, that green stuff. That's called a mold. Of course, we know that, right? That's a filamentous fungus. Yeast are single cellular. We need microscopes to see them. So what do I want you to know from the slide is this one here. Most fungi in nature, they are saprophytic. Don't forget this. What that means, they decompose organic material. Think about that mold growing on fruit or bread. What they're doing, they're using organic material. They're recycling them in the environment, all right? Few are pathogenic. In fact, very few species relative to bacteria. Of fungi, they are pathogenic to humans. But the major thing I want you to know right now, in general, they function in nature, most of them, as saprophytic organisms. They decompose organic material. Viruses, let me just say, viruses are not microorganisms per se. They are microscopic, but they're not cellular. In other words, they're not like E. coli. They are all obligate intracellular parasites. I call them molecular parasites. They can't survive on their own. They require living cells to grow. All right? So that's a very general overview of the groups of things that we'll be studying later on. What I want to focus on today really is microscopy. And this is what I was confused about because in the laboratory we did a little bit about microscopy too. All right, so nonetheless, let's look at microscopes. All right, so this is the most important aspect or thing about today's lecture, chapter 3. Now, some terminology. We all know what a meter is. Here's a meter rule. We know what that is. We know what a centimeter is. This is 1/100th of a meter. So it's 10 to the minus 2. All right? And if it gets smaller, basically, you guys can see those two little marks on that centimeter area. It's a millimeter, right? Of course, you can see a millimeter with your eye. But think about it. Here's that millimeter. I go maybe half a millimeter and smaller and smaller and smaller. Then you can't see anything. You can't resolve anything. So let's look at some other measurements. A micrometer or a micron is exemplified by this sign. That's a mu sign, mu m. What is a micrometer? A micrometer is 1/1,000,000th of a meter. Bacteria is in this range. So Staph aureus, for example, that's a very tiny cell. The diameter is 0.5 millionth of a meter. And then we have this other scale, a nanometer. All right? So a nanometer is 10 to the negative 9th of a meter. This, in other words, this is 1/1000000000th of a meter. Let me just say viruses are in this range. All right? Viruses in the nanometer range, typically between about 40 to about 100. And the larger ones could be a little, maybe about almost 200 to 400 nanometers. So that's pretty small. And then, of course, we have this little angel sign right here, that A with a little circle above it. That's called an angstrom. And angstrom is 10 to the negative 10. This is 1/10000000th of a meter. So I have this in red here. This is where your microorganisms lie. Microns for bacteria, nanometers for viruses. Right? Okay. Now, this is one way to look at them. Here's a nice little scale for you. You get into the angstrom, 10 to the negative 10. This is 1/10 billionth of a meter. You have molecules that's in this range. You get into the nanometer range, you see viruses here. Microns, you see bacterial cells. And then, of course, as you get larger and larger back here, we see parasites. Think about tapeworms. Tapeworms can be about 10 feet long. The largest one is about 30 feet long. That organism is called Diphyllobotrium latum. So these are the worms. All right. Of course, we'll be focusing, for most of our time, we're in this area. Nanometers for viruses, and then microns for bacterial cells. All right. So, again, you should have an inroad into this because last week during laboratories, we did a little lecture on microscopy. So I want to just review a few things. In fact, we're going to see microscopy in the laboratory this week again. So we talked about gram stains last time. We're going to be doing that stain actually next week. We talked about simple stains and negative stains, if you can remember that. We'll be doing that in the laboratory this week. All right. So microscopes. Well, we know microscopes magnify things. We get that. But here's the important thing. The most important feature of a microscope is resolution. And I want to be very clear about that. Yes, it's pretty cool that we can magnify objects, but magnification is not the most important characteristic. For example, imagine if I magnify things 1,000,000 times, but I still can't distinguish A from B. That doesn't do me any good, right? But resolution is pretty cool. Good microscopes are able to resolve two different images. And again, you're going to see this in the lab this week. If I have A and B, and again, I'm not looking at human cells. Human cells are really large. You can see that pretty nicely. But imagine you have two very tiny objects, let's just say two Staph aureus cells. The diameter is 0.5 microns. That's 0.5 millionth of a meter. To separate those two cells that you can see them very clearly, you have to have good resolving power. So what is resolution? Resolution is simply this, to understand it. Here's my field of view. I have two objects. I can see them very clearly. All right? So resolution is defined as the ability to distinguish two very small objects and closely spaced objects as separate entities. So here's A and here's B. Yeah, it's only a micron apart, but I can see them very clearly. If I could magnify them and I don't see A and B, I got a problem. So magnification is not the most important feature for microscope. Yeah, you magnify things, you amplify the size, but might not tell you if it's A or B. Resolution is very important. All right, so the most important feature is resolution. Now, I'm just going to go through just a little bit regarding microscopy. Not much to memorize here. I do want to get the stains, and again, we saw all this in the laboratory last week. So a light microscope is also called a bright field microscope. And again, this is the scope we have in our labs. Of course, we have the objective lens. If you recall, I had a picture of a microscope. I said, hey, guys, I had one that was unlabeled and one that was labeled. If you know the parts of the microscope, you're doing pretty good. That's going to help you on your exam, right? Your practicum. So again, we have something called an eyepiece or ocular. These are the objective lens. You put your slide on the stage. You guys have all used microscope before, hopefully. Here's the light source. You focus your specimen and you resolve your image. Notice this, for this microscope, it's about 0.2 microns. That's 0.2 millionth of a meter. That's the resolution. So you can resolve images that's 0.2 millionth of a meter apart. All right? Okay. Another technique is dark field microscopy. All I want you to know from this is this. Clinically, we can use it to identify the organism that causes syphilis. And that's a little fact I want you to remember. So this is a special type of microscopic techniques. Here are patients who have syphilis. This is on the tongue. This is on the lip, this is on the glans penis, and this is on the female external genitalia. It's all the same type of lesion. If you were to pop that, it seeps a clear fluid. You take that fluid, you put it on a slide, and you look at this through dark field microscopy, not light microscopy, but dark field microscopy. This is what you would see. See those little squiggly things? Those little squiggly things are the organism Treponema pallidum. Don't worry about the name. We're going to get to syphilis later on. But again, this is the organism that causes syphilis. And I've seen this once in my life. This goes back to 1985 as a student at Huntington Memorial Hospital. I haven't seen it since. But nonetheless, you see a dark field back here, and these are the bacteria just kind of moving around. They rotate like this, like a corkscrew, moving around the slide. All right, so this is very helpful, this type of technique. Dark field microscopy, all I want you to remember this. It's a pretty cool technique to help you identify, all right, to observe, I should say, the organism that causes syphilis. All right. This other one here is called fluorescence microscopy. And all I want to say about this is we can use fluorochromes. Fluorochromes are chemicals. When you shine UV light on them, they emit a color that you can visualize. You can't see UV light, but you can see these different colors here, right? Now, this is a pretty cool technique in laboratories all over the place. Because imagine if you have a cell infected with a virus or with chlamydia, for example, they require cells to grow. You can actually have a very special dye that's tagged onto something very specific to the pathogen. You add that to the cells. And then what you would see, that would bind to the pathogen, and then they light up. All right? In this case, of course, we're not looking at pathogens. We have three different colors back here. This is a cell, the cytoplasm. The blue thing you see back here, those are DNA, that's chromosomes. And then these green things are spindle fibers. And again, you put all these images together, you have a pretty cool view of what's occurring in the cell. Of course, if you study mitosis, that's exactly what's going on here. So all this slide is telling you is simply this. We can use fluorochromes. These are chemicals that actually fluoresces when you expose them to UV light. And we have different colors for different chemicals. I'm not expecting you to memorize all that stuff. I'm just trying to help you understand there are different microscopic techniques that we can use. All right? Again, here's another technique. It's called confocal microscopy. You notice we can see things pretty clearly here. All right? Because we label them with certain colors. By the way, the scope that we'll be using in the laboratory wouldn't give you these images. All right? You require a fluorescent microscope to see these images. So I want to move on to the last microscopic technique. This is called electron microscopy. All right? Think about it. In this stage, we're using electrons to see. We're actually going to create an image by using electrons. So an electron microscope is not like a light microscope. Here's how it works. Basically, right here, here's an example of an electron microscope. It's called a scanning electron microscope. This is pretty old. This goes back to the 70s, by the way. We have more advanced instruments, but nonetheless, it illustrates what I want you to see. So this is a scanning electron microscope. Notice this right over here. We can resolve images in the nanometer range. Remember with bright field, it was in the micron range, right? 1 millionth of a meter. Here we can see things that are pretty small, 1 billionth of a meter. So here's how it works. I have the instrument here, and then I have a drawing to describe what actually happens. At the very top of the instrument, you see these cords, these electrical cords going right here, right? That's the electron gun. We have these rings, they're called ends, but really they're magnets. And then here's your specimen.